Today, we're diving into a question that is, frankly, at the very heart of computational biology, and it's getting more complex by the day. What exactly is a cell type anymore? For the longest time, we've thought of it as a nice, clean label on a biological map. But what we're finding is that in the single cell era, the map itself is being completely redrawn. Okay, so to really get into this, let's start with the foundation that we're all familiar with, the modern single-cell transcriptomic map. We start with this incredibly rich, high-dimensional scRNA-seq data. I mean, we're talking thousands of dimensions per cell. And we use amazing dimensionality reduction techniques like t-SNE or UMAP to essentially squash all that complexity down into a 2D visualization that we can actually look at. And what emerges are these familiar, beautiful clusters of cells that are transcriptionally similar to each other. Now, here's a distinction that is absolutely critical to make. The visualization, that's for us. That's for our eyes and our brains. Tools like UMAP are fantastic for helping us see the patterns, but they don't actually define the clusters in a mathematical sense. That job belongs to a totally separate clustering algorithm that operates on the original high-dimensional data to partition the cells into discrete groups. So, once we have these algorithmically defined clusters, the real work begins. We have to figure out what they are. And this process, well, we can think of it as running the Annotation Gauntlet. The standard playbook here usually follows a three-step workflow. You start with some high-throughput, automated methods, then an expert biologist has to come in for careful manual curation, and finally, you have to verify the whole thing. When we talk about that first automated step, it generally goes one of two ways. There's the marker-based approach, where you basically feed the algorithm a list of known gene signatures, or you can take a reference-based approach, which tries to match your new data against a huge pre-annotated atlas. And this just gives you a little taste of the diversity of tools out there. You've got correlation-based methods like SchemEp. You've got really clever tools like SNE that try to cluster and annotate at the same time. And then you have the heavy hitters like Liger, which use sophisticated techniques like non-negative matrix factorization to integrate multiple data sets. But here's the problem. Despite all this sophisticated tooling, automation has its limits. A model is only ever as good as its training data, right? So these tools start to fall apart when they encounter incomplete reference atlases or poorly defined marker genes. And they really struggle when they come across a genuinely novel cell type that simply doesn't exist in any reference map yet. And these challenges, they're forcing us to step back and question the entire premise. The whole idea of discrete, stable cell types just doesn't always capture the biological reality, because what we see so often is not a set of boxes, but a smooth continuum of states and developmental gradients. Let's just make this concrete. Imagine a simple developmental process. We start with a progenitor cell. Now, this cell doesn't just magically teleport to its final form. No, it enters a transitional intermediate state as it begins its differentiation journey. And only then does it finally resolve into a stable, mature neuron. The entire process is a continuous flow, a gradient, not a series of discrete, instantaneous jumps. So the question becomes, how on earth do you put a label on a gradient? It's an incredibly difficult annotation problem, because those cells caught in the middle rarely express their own unique markers. This forces us to just label the endpoints, completely missing the richness of the process in between. This very challenge is pushing the whole field toward a really fundamental shift in thinking. We have to move beyond this static, snapshot view of a cell's identity. And this quote here from Camp and Trutlin's work in Science just perfectly captures this new paradigm. It says a cell type or state is also linked to how the cell would respond to an environmental change or other perturbation. It's not just about what a cell is right now, but what it could become. So the idea is that a cell type is defined not only by its current gene expression profile, but by the entire spectrum of responses it's capable of having when you challenge it, when you perturb it. This completely reframes the goal of a cell atlas. It's no longer just a descriptive dictionary, like a simple parts list for an organism. It's becoming more like a predictive manual that can tell us how these cellular components are going to behave under different conditions. The ultimate goal of this new perspective is, well, it's incredibly ambitious. It's nothing less than charting the entire human phenoscape. And what is the phenoscape? I mean, it's essentially the complete map of cellular potential. It's the comprehensive landscape of every possible cell phenotype that can be reached under every possible perturbation.
Building something like this is a monumental task. It requires integrating data from primary tissues, things like the human cell atlas, with scalable, high-throughput experiments in model systems, like organoids, where we can actually perform these perturbations at a massive scale. And critically, these perturbation experiments can't be done in a vacuum. A cell's potential, its response to any stimulus, is deeply, deeply dependent on the context of its microenvironment and the signals it's getting from all of its neighbors. So the big takeaway here is that a cell type is slowly becoming less of a noun, a static thing with a fixed label, and much more of a verb. It's an entity defined by what it can do. And this leaves us with a final, pretty provocative thought. If we actually succeed in mapping the entire human phenoscape, we could move beyond just classifying the building blocks of life, and maybe, just maybe, we could begin to predict their future.